Hello everyone, welcome back for one last video in Chapter 13 for Intro to Biotechnology. We've talked a lot about PCR, in vitro DNA synthesis, and a lot of other good things. This last video we want to focus on something that is very important, which is DNA sequencing. The ability to be able to determine the nucleotide sequence of an unknown piece of DNA. So way back previously in the semester, we've obviously talked about the enormous implication and significance of the Human Genome Project, which sought to and succeeded in sequencing all of human DNA. And it really just allows us to have a very good and very strong genetic understanding of all human proteins like insulin, which seems to be kind of the example that we've been sticking with throughout the rest of the semester. So the principles that are behind DNA sequencing are actually fairly similar to PCR, especially in the fact that it's going to require all the different things to be mixed up in a tube, and that includes the template DNA that you're wanting to sequence. You're going to need primers. You don't necessarily need both forward and reverse primers, although certainly it can help. And you are definitely going to need DNA polymerase and free nucleotides. Now, the free nucleotides is where we really want to focus our attention here, because this is going to be where things are different. So the modern form of DNA sequencing is what we're going to call interrupted DNA synthesis, because of a very special type of nucleotide that we are going to use called dideoxynucleotides. So what makes a dideoxynucleotide different from your typical deoxynucleotide? So normal DNA nucleotides contain a free hydroxyl group at their 3' end. So if you recall way back to, what was it, Chapter 4 when we discussed all this, a nucleotide has a phosphate group at its 5' end and a hydroxyl group at its 3' end. So when you're talking about synthesizing DNA from 5' to 3' direction, you're talking about a 3' hydroxyl forming a bond with the incoming 5' nucleotide. So that's what normal nucleotides look like. Normal nucleotides do have the 3' hydroxyl group that you can see right here. We call it 3' because it's stuck to the third carbon on the carbon chain here. Dideoxynucleotides are special because they have had that 3' hydroxyl group removed. So if you incorporate a dideoxynucleotide into a growing strand of DNA, you'll be able to do that. You'll be able to get the phosphate, the 5' phosphate to stick onto the 3' hydroxyl group of the last nucleotide on the growing strand, but you're not going to be able to add anything beyond that because nothing is going to be able to attach to the 3' end of the dideoxynucleotide because there is no hydroxyl group there. So once a dideoxynucleotide gets incorporated into a growing DNA strand, you're done. You can't grow the strand anymore. So the DNA sequencing reaction is going to contain, as we said, the template that you're trying to sequence, DNA polymerase enzyme, free DNTPs, your typical normal deoxynucleotides, and then a very small amount of the special dideoxynucleotides. The way that we're wanting this to work is that we should end up, as the DNA polymerase does its job, we should end up with a diverse mixture of different PCR products that we're eventually not able to finish because they, at some point or another, incorporated a dideoxynucleotide. The dideoxynucleotide incorporation may happen right away, it may happen part of the way through, or it may happen towards the very end. We want there to be enough template and enough dideoxynucleotide to be there to ensure that we get every possible length of DNA that is possible. We want to get some DNA that has the dideoxynucleotide at the very first possible position, and then every position after that, so that we can know exactly which nucleotide was supposed to go there. So the 4 different dideoxynucleotides that correspond to adenine, thymine, cytosine, and guanine are also going to be differentially tagged with different, usually fluorophoric substances that will allow us to determine which one of those dideoxynucleotides caused the synthesis to stop. So we'll know exactly whether a adenine was supposed to go here, a thymine was supposed to go here, or et cetera, et cetera. So if we were to load the sample onto a gel, we should be able to see which DDNTPs cause synthesis to stop in which order based on the size of the fragments. Obviously, the smallest fragments represent dideoxynucleotides that were incorporated very, very early on because you would end up with very, very small products. And then the larger fragments represent dideoxynucleotides that were incorporated much, much, much later on. So DNA sequencing really does not become very accurate until you've gotten about 50 base pairs past where the sequencing primer binds. So even though technically you can get some dideoxynucleotide incorporation into those first 50 base pairs, you're not really going to get good accuracy until about 50 base pairs past where the primer binds. So if you know that you're trying to sequence a particular part of a DNA, you want to design a sequencing primer that binds at least 50 base pairs upstream in the 5' direction of what you are trying to sequence. So once a gene is sequenced for the first time, the sequence is usually going to be deposited into a variety of different online databases to make those sequences available to everyone. We've already explored things like this, these web tools for using this DNA sequencing information, such as what you find on NCBI much earlier in the semester. So when you sequence a piece of DNA, you're obviously going to want to know where it came from, especially what organism it came from. To do so, you can use basic local alignment search tools like BLAST to basically tell the database that, okay, I've got this particular sequence of DNA, and then you can see how well it matches up with sequences that have already been deposited in the database. So if you say sequence DNA that comes from a brand new organism, then you can put it into BLAST and you can see how similar that DNA is to other organisms on Earth that have already had those genes sequenced. So that can be a very powerful tool. So for example, you could use a tool like this to blast a human gene like insulin, and you could, just for curiosity's sake, you could see how similar is human insulin to the insulin that comes from dogs or cats or cows or something like that. So your results are usually going to look something like this. So you're going to see all these different genes that kind of pop up as matches for your blasted sequence. So you're going to get things like scores, query coverage, basically how much of your DNA matches what is in these DNAs here. You're going to get an E value, and typically the lower that number is, the better a match you get. So you'll get a pretty good idea of what is going to be the best match based on kind of what tends to be sorted up towards the top of the search results. All right, so that is going to do it for this video. Thanks again, and for this semester, we should have just one more chapter to get through in Chapter 14, so definitely be on the lookout for those videos, and I will see you then. Bye-bye.